In the last video, we looked at the complement system. Part of the role of activation of this system is to recruit cells to the site of inflammation, particularly neutrophils, and to coat microbes in C3b in the process of opsonization. Opsonization is essentially the immune system's way of sticking a warning flag on the surface of the pathogen, telling the immune system that it needs to remove the threat. In the first part of this video, we're going to take a quick look at this process. And then in the second part, we are going to look at how the innate immune system is able to identify what sort of response it needs to mount. The phagocytosis comes from the Greek phago, meaning to eat, and the two main cells that do this for the purpose of destroying pathogens or infected host cells are the neutrophils and macrophages. Macrophages, whose name literally means big eaters, can be tissue resident or recruited from the circulation, while neutrophils must be recruited from the circulation. Both these cells can bind directly to a pathogen, but the process is dramatically enhanced when the microorganism has been opsonized. You can think of this in a way like dipping food into sauce to make it more appetizing. Indeed, in ancient Greece, opson referred to the delicious side dish of any meal. When we looked at the complement system, we identified that some of the cleavage products, especially C3a and C5a, are capable of recruiting cells to the inflammatory site and activating them, while C3b acts as an opsonin. Neutrophils and macrophages both express a receptor on their surface that allows them to bind to C3b that has coated the microorganism, as well as receptors that recognize the Fc region of antibodies. If a pathogen is opsonized by both complement and antibodies, then phagocytosis is even more effective. Interestingly, C3b on its own is not sufficient to initiate phagocytosis. Binding of C5a to its receptor, also on the surface of neutrophils and macrophages, is required. When antibodies or complement proteins bind to their receptor, a signaling cascade is initiated that results in the cell membrane forming feet-like projections that we call pseudopodia. This involves a cytoskeleton rearrangement that engulfs the pathogen, creating an intracellular organelle called a phagosome. Once inside the neutrophil or macrophage, the phagosome binds with a lysosome to form a phagolysosome. The lysosome releases its content into the phagosome, reducing the pH and releasing enzymes that break down the pathogen. Degradation can be either oxygen-dependent or oxygen-independent. The oxygen-dependent pathway which occurs in neutrophils involves a complex called NADPH oxidase, which converts molecular oxygen into reactive oxygen species, or ROS. These include superoxide, hydrogen peroxide, and myeloperoxidase. By activating a halogenation system, hypochlorite is produced, which is essentially a biological bleach. These reactive oxygen species are highly toxic to microorganisms, and superoxide is also important for generating reactive oxygen intermediates, which also contribute to pathogen destruction. Macrophages also use reactive nitrogen intermediates as part of their cell-killing mechanism, combining nitric oxide with superoxide anion to form peroxynitrite. On the oxygen-independent side, which happens in both macrophages and neutrophils, lysosomes release their content into the phagosome, which contains numerous antimicrobial peptides, including lysozymes and cationic peptides, such as defensins, that can form pores in pathogens, as well as enzymes that degrade important microbial biomolecules like elastases, lipases, and nucleases. What happens next depends on the cell. In neutrophils, the cellular debris is released into the local environment via extracellular, where it can enhance the inflammatory response. Neutrophils also have a final party trick after they have phagocytosed a bacterium. Following that phagocytosis, neutrophils will die, and as a final act of defiance, they can produce neutrophil extracellular traps, or nets, where the neutrophil throws out its DNA to trap and help kill further bacteria. In contrast, macrophages, which belong to a group of cells called professional antigen-presenting cells, express part of the broken down pathogen on the surface of the cell in a molecule called MHC class II, and we will learn more about why this happens when we look at the adaptive immune response in more detail. As well as initiating phagocytosis, binding of complement components and/or antibodies can activate the cells to release cytokines and chemokines. And as we've already discussed, these play a fundamental role in shaping the propagation of the inflammatory response and ultimately clearance of the pathogen. So that is the process of phagocytosis, and leads us nicely into the last part of this video, where we are going to look at how the innate immune system can identify that there is a threat and mount an appropriate response. The receptors involved in phagocytosis are part of this process, but there are further receptors that we need to look at. Now, we said that the innate response is the same every time a pathogen is encountered. But that is a bit of an oversimplification, because there is variability in the type of response that is mounted. So while the response to a gram-negative bacterium is invariant, the response to a virus would be very different. This selectivity of response is driven by a series of receptors called pattern recognition receptors, which can recognize two main classes of molecular patterns. Those found on microbes, which we call pathogen-associated molecular patterns, or PAMPs, and those that are released from damaged self cells, which are not normally seen by the immune system, which we term damage-associated molecular patterns, or DAMs. Let's have a look at PAMPs first. There are several different families of receptors that allow the body to identify the nature of the invader, and to a degree tailor the innate immune response. The first family we're going to discuss are the toll-like receptors, or TLRs in humans. This is a family of 10 different single-pass transmembrane receptors with an extracellular leucine-rich domain, which form homo or heterodimers, and are expressed on a wide variety of different cell types, including macrophages and neutrophils, where they contribute to phagocytosis, but they're also expressed on dendritic cells, allowing for a link to the adaptive immune response. Importantly, they are also expressed on non-immune cells like the endothelium and stromal cells, allowing for an early warning of an invader. They identify patterns that are conserved within pathogens, often ones that are central to their survival and so cannot be easily modified. We aren't going to talk in great detail about every single combination of TLR dimers, but to note, all the TLRs apart from TLR3 signal via MyD88 TRAF6, and TLR3 being the exception signals via TRIF TRAF6. But a downstream effect for both is the upregulation of gene transcription via AP1, nuclear factor kappa B, and IRF3. And this helps produce defensins, inducible nitric oxide synthase, chemokines, cytokines, and in the case of the TRIF pathway, type 1 interferons, which are active against viral infections. Note this is a simplified overview of the signalling pathway and does not include all the different molecules involved. Toll-like receptors can be extra or intracellular, where they form on endosome membranes. And when you think about the nature of pathogens, this makes sense. Bacteria are normally extracellular pathogens. The toll-like receptors that respond to bacterial PAMs are found on the surface of the cell, whereas viruses need to infect a cell to hijack the cellular transcription machinery, and so it makes sense for TLRs that recognize viral PAMs to be intracellular. If we look at the extracellular receptors first, they need to be able to respond to both gram-positive and gram-negative bacteria. Gram-positive bacteria have a thick external layer of peptidoglycan, which is rich in di- and triacylated lipopeptides. TLR2 is central to detecting lipopeptides and forms a heterodimer with TLR1 to detect triacylated lipopeptides, while the TLR2 6-dimer responds to diacylated lipopeptides, as well as lipoteichoic acid, or LTA, and zymogen from fungi. TLR2 can also form a homodimer to detect peptidoglycans surrounding bacteria. Gram-negative bacteria have an outer membrane made up of LPS, or lipopolysaccharide, which is recognized by a homodimer of TLR4. LPS is an incredibly immunogenic endotoxin and is one of the most potent activators of the immune system that we know of. TLR5 binds to a feature present on some bacteria called the flagella, recognizing a protein called flagellin, which the bacteria need to move around. TLR10 is a bit of an odd one out, and until recently was considered an orphan receptor, as its function wasn't known. However, emerging evidence suggests it plays a role in detecting listeria, but interestingly also detect viral PAMPs on the influenza virus and seem to be anti-inflammatory, whereas all the other pattern recognition receptors are pro-inflammatory. Now, you may have noticed that we skipped TLR 3, 7, 8, and 9. And this is because these pattern recognition receptors are expressed intracellularly, associated with endosomal membranes. These intracellular receptors tend to recognize nucleic acids from bacteria and viruses, but also have a role to play in autoimmunity by detecting self-DNA that has escaped the nucleus. TLR3 recognizes viral double-stranded RNA, small interfering RNAs, and self-RNA from damaged cells. TLR7 recognizes viral single-stranded RNA, so it's important in the defense against COVID-19. TLR8 can respond to viral and bacterial RNA, while last but not least, TLR9 binds CpG DNA, which is unmethylated DNA found in viruses and bacteria. As noted, location is important with pattern recognition. Our own DNA can trigger an immune response if it is found in the cytosol of a cell, as it should be contained within the nucleus. So just to recap, toll-like receptors signal either via MyD88 dependent or a TRIF dependent pathway. Those that signal via MyD88, which is every toll-like receptor except TLR3, can result in an antibacterial, antiviral, and in some cases antifungal response through the production of antimicrobials like defensins, inducible nitric oxide synthase, and cytokines and chemokines that drive a pro-inflammatory scenario. TLR3 signals via TRIF and results in the production of type 1 interferons, so is heavily involved in the antiviral response. As well as the membrane-bound TLRs, humans have cytoplasmic pattern recognition receptors that help in the fight against viruses, in the form of RNA-sensing RIG1 and MDA5 that interact with the mitochondrial antiviral signaling protein MABs to promote type 1 interferon transcription, as well as TNF-alpha and interleukin-6 release. Additionally, there is a cytosolic DNA sensor for cyclic GMP-AMP synthase, or cGAS, which is also capable of inducing type 1 interferons via IRF3 signaling, or TNF-alpha and interleukin-6 via NF-kappa B signaling. The final group of pattern recognition receptors we are going to look at briefly are the NOD-like receptors, or nucleotide binding oligomerization domain-like receptors. These can detect both PAMPs and DAMPs and can broadly be divided into 4 families. But we are only going to mention the NLRC and NLRP subfamilies, which have caspase and pyrine domains respectively, and are important in the formation of a subcellular multimeric structure called the inflammasome. In the example shown here, we are looking at an NLRP3 inflammasome, forming this wheel-like structure. One of the main functions of the inflammasome is the activation of interleukin-1 beta, which is a potent pro-inflammatory cytokine that is capable of causing tissue damage. So it is produced in a proform that needs cleaving by caspase-1 in order to become activated under the right conditions, and it plays a central role in the inflammatory cascade as we have seen previously. The NLRP3, when it oligomerizes, is capable of cleaving pro-caspase-1 into active caspase-1, which in turn is able to convert pro-interleukin-1 beta into active interleukin-1 beta, which can be released into the surroundings. The NLRP3 inflammasome can also activate interleukin-18, which is another pro-inflammatory cytokine that drives interferon gamma production, and may result in a process called pyroptosis, which is a highly inflammatory form of cell death that sometimes occurs during infection with an intracellular pathogen. And that leads us nicely onto the last part of this video. So finally, we need to discuss damage-associated molecular patterns, or DAMPs, which are sometimes called danger signals or alarmins. DAMPs are endogenous, so they originate within cells and are released in response to damage, and this triggers a non-infectious inflammatory response, or can enhance an infectious response in the case of pyroptosis. It is often the combination of PAMPs and DAMPs activation that leads to an effective immune response, as described by Polly Matzinger in her danger model, that states that the immune system only responds to foreign invaders if they are or have the potential to cause damage, which is the stranger danger response. So what we can see here is an immature dendritic cell binding to both a PAMP from an invading microbe and a DAMP from a cell that is undergoing inflammatory cell death, maybe via necrosis or pyroptosis. The danger model suggests that the effect of these two signals in combination triggers the immune response, and the immature dendritic cell will become activated, mature, and migrate to a lymph node where it can activate the adaptive immune response. A lot of the DAMPs also bind to toll-like receptors and nod-like receptors, although there are numerous specific receptors that are beyond the scope of this video. Examples of DAMPs include the release of interleukin-1 alpha, ATP, self-DNA or self-RNA if found in unexpected cellular locations, and the contents of granules from neutrophils and macrophages, amongst other things. So what is the key take-home message of all this? Well, pathogen and damage-associated molecular pattern receptors allow the body to identify what the invading pathogen is, and we focused on the differential response between a bacterial and viral response in this video, but the body can also detect fungal and parasitic infections. This is important to allow the innate immune system to start to fight and contain the pathogen, activating appropriate cellular responses and helping to activate the adaptive response. So now we've covered most of the key components of the innate immune system, and as you are hopefully starting to appreciate, it is quite a complex system with many moving parts, all working together to produce and resolve an inflammatory response. In the next video, we will look at this process of acute inflammation as a way to try and give an overall summary of the innate immune response in time and space.